Anybody heard of a TED Talk? Who's heard of a TED Talk? Great. So TED Talks, technology, entertainment, design. And these are talks designed to change the world. So you're supposed to, if you get invited to give one of these TED Talks, you're supposed to give a 10-minute talk that will change the world. And there are all these kind of independently organized TED events. So other people, you don't have to be invited to the big conference, but you could have a smaller satellite conference. And there is one that's going to be organized here, right here at NYU. And there's an interest meeting at 3 o'clock on Saturday, November 5th in Tisch Hall. Frank here is one of the organizers. So if you're interested in finding out about it or interested in giving a TED Talk, please see Frank. Could you say, remind us what the theme or what the thought of the theme is?
The theme for TEDx NYU is NYU in the community.
Right.
So talking about kind of blending all of NYU into one community as opposed to having it as a segregated.
Great, NYU in the community. And that's a really great segue to another announcement, which is I happen, after he came up to me in class and gave me this, I happen to be giving a TEDx talk myself on Saturday at TEDx in Orlando. And I'm going to tell you more about the topic material when we get to our lecture on exercise and modulating memory with exercise. But my TEDx event, organized by TEDx Orlando, which has been going on for several years, the theme is the creative spark. So people from business, people in physics, people in art, people in science are going to come together and discuss creativity from their own point of view. So my talk is actually entitled Exercise, Creativity in the Brain. And it talks about the work that I'm doing looking at the effects of exercise on learning, memory, and cognition in the brain. And it gets to the topic that we'll be starting on Monday, which is hippocampal function. Hippocampus is critical for long-term memory, but really exciting new direction for our understanding of what the hippocampus does is that it not only binds different items and unrelated items in our long-term memory, but it actually helps us put new ideas, new concepts together, which really is the basis of creativity and imagination. And so anything that could help your memory and your long-term memory and your hippocampus may also be able to improve your imagination and creativity. And so I'm going to be talking about that on Saturday in Orlando, and I'll be giving you a preview of, not a preview, but a post-talk of that talk in our talk. I think it's on November 21st on exercise in the brain. I should also say that at the end of today's lecture, I'm going to repost an updated syllabus because we got a little bit off schedule, not off schedule, we just took a little bit longer on the auditory lectures than I had originally scheduled, not a big deal. So I'm going to repost the modified set of lectures so you know exactly what to read and when. So this hopefully should not change for the rest of the semester, the new one that I will publish, that I will put on Blackboard. Okay, so what we want to do is continue our motor discussions and move on to motor diseases. And so I wanted to take a second and step back. At the end of the last lecture, I showed you that video of brain gate, that is the ability to record from the primary motor cortex of humans that are paraplegic, they're shut in, and to allow them to interface with computers. So you saw the woman who was paraplegic being able to move a cursor with her mind. That is called brain-machine interface. And the man you saw interviewed, John Donahue, is a neuroscientist at Brown University, and he did studies just like this, okay, with monkeys, moving different instruments, recording in area V1, trying to figure out what these, sorry, not V1, M1, recording in M1, primary motor cortex, trying to figure out how these neurons are coding for the movement that the animal was about to give. And we talked about the different kinds of tuning curves that an individual motor neuron can have. Here's one type of tuning curve, but we talked about that there's lots of other different kinds of tuning curves, curves that are much more specific, only responding, for example, when the animal moves his arm to this location and nothing else, and others that might be even broader, so a larger, all of these movements to all of these different directions, causing strong activity in terms of action potentials. So what that video showed was actually really, really amazing. It's not just that you can pop a little electrode array in somebody's head and then suddenly they're able to use a computer and move a cursor around. There's a huge step in between, and that huge step has to do with our understanding of this neural code. All of that research and all of that technology that you saw in the video was possible because of these kinds of basic science recordings that we're doing in the monkey primary motor cortex, just trying to understand how it's organized. So what did they do? Actually, they did the experiment that they showed in her in monkeys well before they did it in her. So monkeys got recorded in their primary motor cortex as they were doing different movements, and then they took away the cursor. And then what they did is looking at the post recordings of the patterns of neural activity and what they knew that the animal did with his motor response, they basically decoded what that activity was when he made this kind of move versus this kind of move versus this kind of move. So you were able to predict, based on this pattern of activity I've seen in hundreds and hundreds of cells, what is the animal actually going to do? So they tested this in monkeys. Once they thought they had a good decode of this complex neural activity that you're seeing in M1, then they took this joystick away. And then they had the animal just look at this cursor, and then based on what the animal was thinking, they had the cursor move in what they decoded, where the animal was intending to move. And the animal actually got very good at controlling with his mind where that cursor should be going. But there's a huge step in there. There is recording, decoding of that recording, saying, okay, my translation of that neural code, that pattern of action potentials is, okay, I think the monkey is trying to move the cursor or move his hands to the upper left, like this. Not an easy task. You have hundreds of neurons. Not all of them are as easily decoded as this. Some of them are very broad. Some of them are not doing anything associated with the task. Some of them are very, very specific. How do you understand that? And that's what the brain is doing all the time. So they use that understanding to try and say, okay, let's try and help people that have a perfectly good working motor cortex but can't move their body because they've had spinal cord injury. And so they did what they are able to do with non-human primates, which is put an array in and record. Yes, question?
How do they identify the individual neurons to put the electrode in, and how do they find it?
That's a great question. How do they find the neurons? Because in a normal person, you could just stimulate. I'm going to stimulate right there, and what happens? Oh, my arm moves. So but the woman can't move her arm. So how do you do that? There's a number of different techniques you can use. For example, functional magnetic resonance imaging can be used, and you could ask the person. So fMRI, MRI machines can be used to actually measure the activity in certain brain areas. It's not nearly as precise as this kind of single unit recording, but you can generally map out a hand area. You could ask the woman, think about moving your hand. So there are a number of different techniques, but that's a really good question. So first thing, they have to figure out where is the hand movement area or eye movement area, and which side of the brain may be functioning better in terms of giving a clean signal. So you can find that all out putting the person in an MRI magnet. And then you have to put your array electrode in. So this is not let's just try this today. It's brain surgery on a human. You have to do a craniotomy. You have to open the dura, expose the brain, and then lay on an electrode very, very gently on the brain. You don't want it to damage the rest of the brain, but these are electrodes that are sticking in the cortex. And then you have to get a wire to come out, and that's what the metal thing that you saw on her head was. That was the wire connection for all. It was an array of electrodes, okay? And you can make that nice and sterile. It's actually very similar to what you use in a monkey behavioral physiology situation. And then you have a situation where you can just screw on the connection for that connector for all the electrodes. And every day, you can't move these electrodes, but you get an array of activity in the area of, let's say, the hand. And then you ask the person, okay, get really good at thinking about moving your hand up. Move your hand up. Move your hand up. And the person gets very good at that, and you get good at decoding it. Then you test, move right, move left, and you do enough trials so that a computer can now predict what this pattern of activity is actually associated with. And that's what the computer uses to then decode what the person really wants to do. It's actually cool. If you go and Google BrainGate, you can see all sorts of cool videos of what this company is doing. Not just having the person look at a computer screen and they can do their email, they can type, they can watch TV, they can turn the music on and off, as you saw. But they're now doing things where you can actually control robotic arms. So there is a trial where I think it's the same woman who has a robotic arm, and she is imagining taking that arm, grasping a cup, picking the cup up and putting it down. I mean, if you can imagine being shut in like that for 20 years, the ability to control something, to do exactly what you want, is so powerful. So this is a really, really exciting implication of this basic science work, understanding motor cortex. But I just wanted to give you a feeling for how complicated it is. It is very, very complicated to decode what's going on there. There's not just one neuron saying, okay, straight, left, right. There's a huge array of neurons, and we're still struggling with trying to decode that in the complex way that you and I are able to decode that every day to make all the millions of kinds of movements that we're able to do. Okay, any other questions about brain? Yes?
For someone who has never been able to walk, let's say, like when they were born, they had like a problem, let's say.
Yeah.
Would this still work? Because they wouldn't necessarily know how to walk.
Yeah, that's a good question. I don't think it would work well. I mean, this only works well because she had a relatively well-tuned up primary motor cortex that could do all the normal things. And they were actually shocked that it still worked after whatever, 15, 20 years of being shut in. But it means that, you know, she's still imagining. She still imagines, I'm able to do that. You know, she probably wishes, I wish I could scratch my nose. I wish I could scratch my nose. And, but you're right, it only works because she had learned how to use it with all the parts intact. So I don't think they've ever tried it with somebody who could never walk. And it's not really walking. You have to be able to reach. They're doing most of the stuff with reaching, so they could work with robots and or driving the wheelchair is also a biggie. Any other questions? Brain-machine interface. Okay, very, very cool stuff. So just a little bit more, we talked about what the motor cortex can do in terms of coding so many different kinds of complex movements. But just like the auditory cortex or the somatosensory cortex, these sensory cortical areas that had a lot of plasticity, motor cortex also, primary motor cortex also has a lot of plasticity. So this is a figure from your book illustrating an experiment in which a rat was trained to do very detailed wrist and digit movements. He had to go in and put his little paw in a little tube and use his little fingers to make a very difficult discrimination. It took weeks and weeks of training. And what you can do is map out the wrist and digit area before training. And this is all the area. The blue and the green are the entire brain areas. This is just a slab of cortex. primary motor cortex that was mapped out. And the green illustrates the locations that were, when stimulated, would stimulate the wrist and the fingers. So these are areas representing the wrist and the fingers. But then look what happens post-training. Even after a few weeks of training, you get a huge change in how this primary motor cortex is organized. Not just areas that expand, but they've completely changed their organization relative to these blue areas that I don't know what those are quoting, but a really dramatic change. So, you know, imagine a child learning how to play the violin for the first time, and the plasticity taking place in their primary motor cortex, or anybody learning anything intricate with their hands for the first time. You have an enormous amount of plasticity going on. And you can look at this experimentally, again, just by recording in these areas and mapping out, just like we did in the somatosensory system, just like they did in the auditory system, for looking for evidence of plasticity, training as a function of experience. Okay, now let's talk a little bit about, we talked about M1, primary motor cortex. What about some of these other areas that are associated with M1? So we talked about primary visual cortex, and then visual association areas. Farther and farther areas are doing even more complex things. So the parahippocampal place area and the fusiform face area are higher level visual areas that, you know, take that visual input and really are very specialized for places and faces. Well, just like in the sensory systems, motor cortex has association areas, non-primary motor cortex. The first one I want to talk about, very interesting, is supplementary motor area. And supplementary motor area primarily is on the medial surface of the brain. Here's primary motor cortex just anterior to the central sulcus, okay? What was found just posterior to the central sulcus? What is the cortical area just posterior to the central sulcus? Homunculus of what? For what? Somatosensory, yes, somatosensory homunculus. Because we also have a motor homunculus, remember? So you have to be, if you just say homunculus, you have to tell me whether it's somatosensory or motor. But yeah, I think you were thinking about the right thing, just, and you said it. Posterior to the central sulcus is primary sensory cortex, just anterior to the central sulcus is primary motor cortex. And then if we go medial, remember this is a mid-sagittal view of the brain. You all recognize the corpus callosum here. Here is the thalamus. Here is the superior inferior colliculus. Here's the cerebellum that we'll talk about. Just here on the medial surface is supplementary motor cortex. This area, damage to this area, so damage to primary motor cortex will not allow you to move. You can't move the area that gets damaged. If you have a stroke in this area, this is what causes paralysis on one side, problems with talking. But what about a supplementary motor area? That shouldn't be so bad, right? Actually, it's devastating. Lesions for to the supplementary motor area makes it so you cannot initiate any new movements. So there's no voluntary movement possible. Once you get going, you might be able to move something, but that is a serious problem. So initiation of movement, this area is one of the areas critically involved in initiation of new movements. This area, particularly supplementary motor cortex, is activated when you're imagining complex motor plans. So there's initiation and also kind of elaboration of complex motor plans. These are very, very complicated areas. We do not understand all of the details of these, but I'm just give you some little tidbits to give you a flavor for what these areas may be doing. Okay, so the other supplementary motor area is let's see, premotor cortex. Premotor cortex, as the name implies, is just anterior to the primary motor cortex. So it's the blue area. right here. And the premotor cortex is associated with movement that's guided by an external stimulus. So if I was asked to actually come over and touch this, my premotor cortex would be very much engaged. It's not an initiation of, oh, I feel like walking over here, but I feel like going over and have kind of an object-guided response. That is where the premotor cortex comes in. So it's involved in mapping object-derived movement. And this is very interesting because one sub-area within the premotor cortex that I want to spend a little bit of time talking about is specifically involved in kind of object-based movement and perception. So this is an area called the mirror area of the brain, and the neurons in this area are called mirror neurons. So recording, these neurons were discovered by an Italian neurophysiologist named Rizzolatti, and he discovered them by accident. So what he saw was he was recording in the premotor cortex. So of course you're asking animals to do a movement, and he found that these neurons, as I was saying, were related to when movement was guided by an external stimulus. So when they had to go pick up a raisin, go get it, pick it up. Pick a raisin up. Every time the raisin got picked up, you got firing of this particular neuron. So therefore, this neuron was kind of a reaching for small objects neuron. And he studied these neurons, see whether if I pick up my cell phone, does it respond? No, it's more specific for small little round things, so it's specific for picking up a raisin. But then they had the electrode in the animal's head, and the animal wasn't picking up a raisin, but the experimenter went over and picked up the raisin. And that cell that they had been recorded from started going crazy. The same high level of activity the cell gave when he saw somebody else picking up a raisin was the same pattern of activity that was seen when the monkey himself picked up a raisin. So this cell was not only responding to the movement itself, but seeing the same actual movement. Hence the term mirror neurons. So this is found in an area close to the premotor cortex, an area called F5. F happens to stand for frontal area 5. And here is an illustration of, this is time over the x-axis, and the activity in spikes per second and action potentials per second on the y-axis. So the higher these lines here, the more action potentials. So here's what they first found. Picking up little raisins, every time the animal picked it up, the cell fired a lot. And we thought, okay, fine, find another kind of motor, specific, more complex motor cell. But then, by accident, when the monkeys were watching an experimenter pick up a raisin, same kind of activity. Maybe not exactly the same, but huge activity. And this is much more specific to other things that the animal could do where you didn't see any activity or any activity when the animal was seeing something. So let me go to a really interesting video, because this isn't only about motor function, but we think that these neurons in this memory and frontal lobe area may have something to do not just with learning motor actions, but also with our capacity for empathy and to be able to feel the things that we see on the outside. It's a really interesting idea. So let's just look at this quick video. It's actually a really nice illustration of that. Mirror neurons. Great. Okay.
Hello again. Gaze into a mirror, and what do you see? Well, I see my face, of course. But in my face, I see moods, I see shifts of feeling. We humans are really good at reading faces and bodies, because if I can look at you and feel what you're feeling, I can learn from you, connect to you, I can love you. Empathy is one of our finer traits, and when it happens so easily, perhaps because, and this is brand new science, this is just out of the lab, we may have some special circuitry in our brains that helps us whenever we look at each other. Ask yourself, why do people get so involved, so deeply, deeply involved with such anguish, such pain, such nail-biting tension over football?
Cleveland Browns are gambling on defense.
Why are we such suckers for sports? And it's not just sports. We can lose it completely at the movies, at video games, watching a dance. Is there something about humans particularly, that allows us to connect so deeply when we watch other people? Watch them moving, watch them playing, watch their faces. Well, as it happens, scientists have an explanation for this strange ability to connect. It's new.
It had never been found on the cellular level before.
A set of brain cells found on either side of the head. Among all the billions of long branching cells in our brain, these so-called mirror neurons have surprising power.
What we found is the mechanism that underlies something which is absolutely fundamental to the way that we see other people in the world.
And it began entirely by accident at a laboratory in the lovely old city of Parma, Italy, where a group of brain researchers was working with monkeys. And they were testing a neuron, that's a brain cell, that always made this sound whenever the monkey would grab for a peanut. So the lab had all these peanuts around, and whenever the monkey made its move, the neuron would fire. Scientists thought, now here's a neuron that is essential to motion. It's a motor neuron. Then one day the monkey was just sitting around, not moving at all, just sitting, when a human scientist came into the lab, and when that scientist grasped the peanut, the monkey's cell fired. Now, the monkey hadn't moved. It was the human that had moved, suggesting that this neuron up here couldn't tell the difference between seeing something and doing something. Seeing and doing were the same. Or more intriguingly, that for this neuron, watching somebody do something is just like doing it yourself. The head of the lab, Giacomo Rizzolatti, thought, wow.
The same neurons, one neuron, fire both when the monkey observes something and when monkey is doing something. It's almost unbelievable.
It was surprising because this cell which was involved with motor planning for the monkey turned out to be interested in the movements of other people as well.
Some people call them monkey see, monkey do neurons, but the name that stuck is mirror neurons, because with them, the brain seems to mirror the movements it sees. This accidental discovery got scientists thinking, doing more tests, and it soon became pretty clear that this is not just a monkey thing. It's a people thing, too. We all know that humans learn by looking and copying. That's what infants do. First you look, then you do.
Ready? Let's see your feet this way.
And once you've watched and copied and learned a set of moves, you not only have them in your head.
Look, you put your shoe on. Yeah, she brushed the
If you see somebody else doing it, you can share the experience.
And you want to do it with me?
They know the moves, you know the moves, so you can move with them.
If you can use the years of training that you yourself have done, learning to crawl, then learning to walk, then learning to eat, this is an incredibly rich set of knowledge that you could apply to the problem of actually seeing what's going on.
So that's why when I head down this street carrying all these packages, not only do people watch, look how they're watching. They feel my predicament, because they know what it's like to carry heavy packages. They all know about carrying. So as they watch me moving, they can feel themselves moving. Their neurons are mirroring the action.
Okay,
These neurons may...
great. So you get the idea. Let's see. You get the idea. So the cool thing here is that, you know, we started out with these kind of mechanical brain areas that are important for making movements. But it turns out that just kind of one synapse away from primary motor cortex is this brain area that seems to be implicated not only in all of our motor learning. I think everybody can understand that being able to practice things when you're a baby and to mirror things when you're a baby is a huge part of our learning. But to be able to actually look at somebody and see whether they're interested looking or bored looking or really engaged or maybe not a little bit tuned out, I can see that by the expression that you have on your face. And I could even imitate that expression. So obviously all of you guys are like this because you're very, very engaged. But it's a huge part of our social interactions. So yes?
Sorry, so does that mean that, I don't know if you recent comments, but people who have trouble identifying other people's facial expressions have like miscegenation?
Yeah, that's an interesting question. So the question was, does that mean that people that have difficulty identifying a face have missing mirror neurons? That could be one possibility. There's also a possibility that the face area that is also important for recognizing faces as well as facial expressions could be damaged. The amygdala, an area that we're going to talk about in a couple of weeks, important for emotional processing, is also critical for our ability to identify and differentiate a scared face from an angry face. So there's, in fact, a lot of brain areas that are involved in that problem. So it's not as simple as, oh, you must have a mirror neuron area. But the mirror neuron area is involved in certain aspects of empathy associated with, I mean, he does an experiment a little bit later. I downloaded this for you on Blackboard. So if you watch the rest of the video, you can see him watching in an fMRI magnet. You can see him watching different facial expressions, and this brain area lights up, his mirror neuron area lights up. But then when he actually imitates those same facial expressions, it also lights up. So it's very, very complicated. There's, again, more areas involved, but this is one of them involved. Okay, any other questions about mirror neurons, what they do, what their implications are? So again, this was discovered not that long ago. And again, if you think that everything in neuroscience is already known that's in your book, this was an accident in the lab. We happened to identify, just as I described and then the host there described, completely unknown that these neurons would respond when they saw the same movement as they were responding to the movement itself. So lots of new discoveries coming on, and it has lots of implications for what these brain areas might be doing. Okay, so that is the end of this last lecture. And then we're going to move on to motor diseases. Okay. Is this it? Yes. Okay. So for diseases, I just wanted to, again, review for you the main kind of components of the motor system that we started with last week. We have the main kind of pathways coming from primary motor cortex, going down the pyramidal tracts, crossing over at the desiccation of the pyramidal tracts and going down, and they terminate in the ventral root of the spinal cord. What's found in the ventral root of the spinal cord? What kinds of neurons are those? What's found in the, yes? Alpha motor neurons, perfect, excellent. The final common pathway, those are the neurons that go down and they are causing the contractions of the muscles. Okay, and so here is the primary motor cortex neurons coming down. They will synapse right here in the ventral horn. This is where the alpha motor neurons are, and they're going out the ventral roots and joining the spinal nerve that, as you know, includes both sensory and motor pathways. Okay, so we talked about primary motor cortex, supplementary motor areas. Now just a word on two other extrapyramidal systems. Extrapyramidal are the non-primary motor cortex regions, and that we'll talk about really focusing on one more than the other. The first is the basal ganglia. So where did you learn about the basal ganglia? Have you heard of it before? Anybody? What is the basal ganglia? Did you learn about it in class or in lab? Anybody do a brain atlas? Anybody label the basal ganglia? No? You did? Yes, you did. Okay. Okay, I'm glad that's settled. So you did label the basal ganglia, and we're going to go over it now. And this is exactly why you did this brain atlas, because so you have a better 3D understanding. And these aren't just names that I'm throwing out to you, along with all the other hundreds of names that I've thrown out in this class. But you have a feeling where these structures are. You modeled it in clay. You took a picture of it. You molded your body into the form of it in your human brain twister. So here we are talking about the basal ganglia. The caudate nucleus and the putamen are known together as the striatum. Other parts of the basal ganglia include the globus pallidus, substantia nigra, and subthalamic nucleus. What is the basal ganglia good for? The basal ganglia help determine the amplitude, direction, and initiation of movement. Okay? So we talked about the supplementary motor cortex area also involved in initiation. Basal ganglia is also involved as well as helping you modulate amplitude, how strong do you make a particular movement. Lots of control in things like walking, where you have to keep the amplitudes coordinated with your whole body, and direction of movement, so you have a directed movement. Here is a view from the side of these different structures. The caudate here is right here. The head is over here, and here's the tail coming down. Here is the putamen right here, more laterally, and then medially here, this is inside, is the globus pallidus. Here you see the location of the subthalamic nucleus below the thalamus, and the substantia nigra. So we've heard about the substantia nigra before. What is the major neurotransmitter used by the basal ganglia? Anybody know? Acetylcholine? No, that's close. Another one? What? GABA, no. Another one? Dopamine. Thank you very much. Okay, excellent. So dopamine, which we talked about in our neurotransmitter lecture, is found and synthesized in neurons in the mesostriatal pathway that originates in the substantia nigra. And the substantia nigra is the dopamine kind of hotspot synthesizer that innervates the striatum. And this pathway, the dopamine projection to the striatum and the basal ganglia as a whole, is important in motor control. And neural loss, as we'll talk about at the end of this lecture, is the cause for Parkinson's disease, quite a common motor impairment. Okay, so here is, again, these last two slides were simply taken from your lecture on neurotransmitter systems. They should be familiar to you. Here is the origin of the mesostriatal pathway. This is the location of the substantia nigra within the brainstem going up, and these neurons in the substantia nigra are dopaminergic neurons that are spitting out their dopamine at the level of the striatum. Okay, so that is the basal ganglia, important for amplitude, direction, and initiation. Cerebellum is the other major structure. We're not going to talk too much about this. You all saw the cerebellum in your brain dissections. Anything strike you about the cerebellum, just the look of the cerebellum from your brain dissections? What does it look like? Any thoughts? Yes?
It kind of looks like it's going to fall off at any second.
Okay, it looks like it's going to fall off at any second because it's heavy. It's very, very invaginated with lots of gyri. Cerebellum means little brain. There's more neurons down here than there are in your cortex. So it looks like it's going to fall off because it's filled with these neurons and it's just hanging on there for dear life. Okay, cerebellar cortex contains the Purkinje cells that I believe. Did you look at a Purkinje cell in your microscope sections? Yes? Okay, good. So what did the Purkinje cells look like? Anybody remember? Yes. Big. They're huge. Exactly. They're huge cells. Oh, I should have. From our very first and second lectures, there were beautiful drawings that I showed you of Purkinje cells that both Ramon Cajal and Gehrig made. So go back and look at those beautiful drawings. They are humongous. They are some of the largest neurons in the brain, and they make up the major cell type of the cerebellar cortex. Purkinje cells are negative. Cerebellum is important for control of skilled movements. So all of these small skilled movements that you are learning over your lifetime, especially when they become automatic. Texting is a very cerebellum-dependent activity, so people with cerebellar lesions will not be able to text nearly as fast, and they can't win that texting competition where you can text 200 characters in a minute or some amazing thing. Not possible without the cerebellum. But the only other thing that I do want you to learn, to know about this, and this is described in the book quite nicely, is that all the Purkinje cells are inhibitory. So all this control is done through inhibitory control of other areas. And actually that's very similar to what we're going to see in the basal ganglia system that we'll go over in greater detail. Okay. So those are some of the systems, the pyramidal systems, premotor cortex, supplementary motor cortex, some of these extrapyramidal systems, including the basal ganglia, cerebellum, lots and lots of brain systems you should be telling yourself that are helping me move around in my environment, that are helping me do the little small tasks as well as the big tasks that I have to do from a motor perspective. So now let's talk about all the different ways these systems can go awry in damage, effects of damage first to the spinal cord, and then effects of diseases to different parts of the motor system. So we're talking about simple, not simple, but very, very common spinal cord injuries, and it's actually very sad. Most of the spinal cord injuries, and there's about 10,000 new traumatic spinal injuries each year. Most of those 10,000 spinal cord injuries are happening to people about your age. Why? Because you're active, because you're driving, because you're doing things like, you know, those cheerleaders that jump up and then flip around and get injured. Sorry, not all 10,000 are cheerleaders get injured. But there's most of these are young people. Why? Because you're active, you're doing things, you get in car accidents, and the result of that is spinal cord injury. And if the injury is to a large enough extent of the spinal cord, you get what is called flaccid paralysis, which is reflexes below the level of the injury are lost. So if you have a higher up spinal cord injury, you can't even get the simple knee reflex because the damage is so severe. If you don't have that much damage, it might be even worse because you do get reflexes, but the reflexes are completely uncontrolled. So you have just spastic movement between your muscles and your spinal cord, but no control from your upper motor cortical areas. So very, very bad news. But the exciting thing is that all the research that was done to identify the pathways, where they go, how they work, is all going to help try and improve and cure spinal cord injuries. This is a huge area, and one of the, these causes become galvanized behind a particular person. And for a long time, spinal cord injury spokesperson was Christopher Reeve. You all know him as an actor. Everybody knows who Christopher Reeve is, right? Yeah. So he was injured in a horse riding accident at his neck, so he became completely paraplegic. But instead of just sitting back and not doing anything, he became such a strong advocate for finding new cures and for research, doing research specifically directed at finding out how to get that spinal cord healed. How come those neurons that get injured, why can't they grow back across the injury again? Why can't they find their connections that they had before? And those are the kinds of causes that he was really passionate about. I actually had the privilege of seeing him speak at the Society for Neuroscience meeting, which is the largest meeting for neuroscientists in the world. It's an international society. There are something like 25 to 30,000 neuroscientists from around the world that come to this annual meeting. It's going to be in 2 weeks in Washington, D.C., and that's why in 2 weeks on Monday you don't have a lecture, because I'm going to be at that meeting. But an amazing program that they have, that they started, I don't know, maybe 7 or 8 years ago, is a program called Neuroscience in Society. And they ask non-scientists to come and address the Society for Neuroscience. And the first person they had come and address the society was Christopher Reeve. He came in his wheelchair. He had a breathing tube because his actual parts of the brainstem that were important for breathing were also damaged. But he was a very, very powerful speaker. And of course, he was speaking to the converted. We are all for neuroscience research, but it was a privilege to actually see how strong his message could be, not to a scientist, but to everybody else who could support monetarily, but also just theoretically, the idea of what can we do to help spinal cord injury. It is a solvable problem. So what he, one of the many things that he helped generate was funding for these kinds of research strategies that are being used today to try and heal the spinal cord. So let me go here first. So here's the problem. We have the spinal cord that we've been talking about. Spinal cord has all the sensory inputs coming in, has all the motor outputs coming in. The motor information is coming from primary motor cortex. There's lots of synapses on these primary motor neurons. But the problem is that if I have damage, here's the damaged region of the spinal cord, all of the pathways going down lower to this region, they get cut off. And these neurons, these axons degenerate. Let's say I have a primary motor cortex neuron up here, and it's going down here, okay? I have damage to this part of the region, and my axon just withers away, okay? It dies. It can't reach this area anymore, and that's why I cannot control any of my movement lower than this damaged region. How do we cure that? How do we fix that? This is like a mechanical engineering problem. We have a damage to one part of a pathway. Can we just patch it up? Okay, so here are some of the things that people are looking at. Stem cells. Who knows what stem cells are? Yes, what are stem cells?
Basically, they take some of the cells like from early in like the fetus stage.
Yes, in the fetus stage.
Before they get like determined to be a certain type of cell.
Exactly.
And then they put it somewhere like someone's that's grown body, and it kind of becomes the cell where they put it.
Exactly. So these stem cells come from fetal tissue, and they have the potential to differentiate into any kind potential. They have the potential. They don't necessarily do it, but they have the potential to differentiate into any possible cell. And the idea is you put it in an area that is damaged, and all the factors and all the inputs in that area could help those stem cells actually differentiate into the kind of cell that you need. So you can grow your own cells in theory. That is why the stem cells are so fascinating to neuroscientists studying neurodegenerative diseases. It doesn't solve the whole problem, even if I have a whole plate full of hippocampal cells that have degenerated in Alzheimer's disease, how am I going to get those cells into the hippocampus to connect up to the right places? That's still a hard problem. But for spinal cord injury, the idea, it's still a hard problem. I get stem cells to differentiate into motor neurons and get those neurons to actually cross the damaged region. Very, very difficult problem, and starting with the problem of how do I get the stem cells into the kind of cell that I need. But luckily, there's more than one answer. Second, you can transplant glial cells to promote regeneration. You remember those glia cells? We only spent literally 1.5 minutes on them. But one of the goals of these glia cells is they clean up debris, but they also form kind of glia pathways to help axons grow in a particular direction. And so what you can do is put the right kind of glia cells there to help promote the damaged axon to grow over this region. It turns out that these glia cells are good at that. And they're good at that because part of what they bring is neurotrophic factors. Neurotrophic factors are chemicals in the brain that the brain synthesizes these endogenously, and they help things like learning and plasticity. A lot of neurotrophic growth factors are present in development. As lots of neurons are being born, they're going to new places. You need these trophic factors to get the neurons to the right place, but they're actually also active in adulthood. And the idea is that maybe a combination of neurotrophic factors and adhesion molecules with the glia can help the damaged neurons grow. So you have a trophic factor, grow in the right direction with adhesive factors, and grow in the direction with the help of glia across this damaged area. And then another strategy that has been used is, okay, we have a damaged area here. Let's just try and transplant a peripheral nerve and bypass this area. It's like a heart, it's like a valve, heart bypass surgery. This is a nerve bypass surgery. Think about how small that space is in your spinal cord. It is surrounded by your spine, your bone. Not an easy area to manipulate. It's not like the cortex where if you do a craniotomy, you can see the cortex laying right there. These are just give you a flavor for some of the ideas that people have used to try and improve this situation where thousands and thousands of people are affected every day. And there are some improvements and some hope that some of this works, but we're still a long way from the neuroscience of it. But there's lots of hopeful strategies as indicated here. Okay, so other disorders, not of just the spinal cord damage, but disorders of motor neurons. Poliovirus, we've all been vaccinated by polio. This was a huge problem in the 1950s. A virus very, very contagious that destroys motor neurons on the of the spinal cord and brainstem. So most commonly, polio would leave you unable to walk. But most severely, if it gets up into the motor neurons of the brainstem and the breathing-related areas, because those are controlled by motor neurons, you end up in these iron lungs. And I don't know if you've all seen this. This is a picture from the 50s of a whole ward of people with polio that have to be in these iron lungs because they couldn't breathe on their own because the polio went up and it affected their motor neurons in the brainstem. Okay, but thank goodness we have a vaccine. This is the vaccine that was invented by Jonas Salk. He has an institute in San Diego. I actually did my graduate work at the Salk Institute, a beautiful institute where I would see him walking around, contemplating new scientific ideas right next to the beautiful Pacific Ocean. And so he was the one that came up with the virus that actually, the vaccine that actually was successfully able to eliminate polio almost completely. There's still small outcroppings, but major improvement. Okay, second disorder of the motor neurons is amyotrophic lateral sclerosis, ALS. And that is another form of damage, death of motor neurons of the brainstem and spinal cord. It's also called Lou Gehrig's disease. And I didn't put this on yet, but I'm going to put a really touching essay by a former NYU professor, Tony Judt, who is a beautiful writer, and he suffered from Lou Gehrig's disease. He died a few years ago. Brilliant, brilliant man, but at the end of his life, he was able to maintain his speech, and so all he could do was he worked on books, he worked on articles, but this is a beautiful essay about what it's like from his perspective to get to the worst part of his day, which was nighttime, where he was kind of wrapped up like a mummy and left alone, and he could do nothing but think and contemplate. And it's actually a beautiful essay. So I'll put that up at the end of the lecture today. ALS is not that common. There are inherited forms of ALS, and there are different forms. Some of them will cause death because you get the complete degeneration of those motor neurons that control your breathing, and then you can no longer survive. But some forms allow you to live for a long time. So Lou Gehrig had a very fast form. He was a very famous baseball player, as you might know, and very quickly succumbed to ALS. Stephen Hawking has, the physicist, has ALS, and, but he has survived for many, many years. He had it as a child, and obviously not affecting his cognitive abilities at all, but has very severe motor disabilities. And maybe you might not know that Mao Zedong died of ALS as well. Other forms of disorders of muscles are muscular dystrophy, which you might hear of. This is a disorder that leads to degeneration of the muscles themselves. So the nerves are fine, but the muscles degenerate because of the lack of a protein that is, this is a genetic disorder, and this protein is not present, and it causes gradual disintegration of your muscles. Myasthenia gravis is an autoimmune disorder where patients develop antibodies to their own acetylcholine receptors, including the acetylcholine receptors in all of their neuromuscular junctions. So what would that mean if you had antibodies against killing all of your acetylcholine receptors in your neuromuscular junction? What would that result in? Yes?
You said that the antibodies belong to kill all the receptors as opposed to the acetylcholine, is that what you said?
Yes, all the antibodies kill all the acetylcholine receptors at the neuromuscular junction. Right, acetylcholine would have nowhere to go, and then what would that result in terms of what a person would be able to do or not? That's right. They wouldn't be able to send out any message at all. It would be like you're completely paralyzed. If you, I mean, think about this. Think about all the different pathways and what would happen if you eliminated this part of the pathway, that part of the pathway. So if you eliminated a spinal nerve, right, you would eliminate motor function to a particular area. But if you eliminate all acetylcholine receptors in all muscles, you would not be able to move at all, okay? So the scary thing is, anybody ever try pufferfish in a Japanese restaurant? What happened? Did you survive? Yeah. Okay, good. Did you feel anything funny? Okay, so pufferfish is, has tetrodotoxin in it, which is a toxin for acetylcholine receptors. And the famous chefs in Japan that prepare the tetrodotoxin fish, the pufferfish, give you enough tetrodotoxin to give you tingles. So, but you know, it doesn't kill you. Luckily, obviously, they didn't kill you. So, bad news to damage your, or to mess around with your acetylcholine receptors. Okay, so fine, good. So in the last 15 minutes, we're going to focus on a very common form of disease, this time of the supplementary motor system, a disease associated with the basal ganglia, again, caudate, putamen, globus pallidus, subthalamic nucleus, and substantia nigra. Remember, the dopamine is coming from the substantia nigra and sprinkling dopamine all through the caudate and the putamen. Okay? Here is a cross section. If I take a coronal section right through here, I will see, here's the lateral ventricle, here's the caudate. It's a smaller caudate, so we're a little bit more posterior. Here's the putamen on the side. Here are two aspects of the, two sections of the globus pallidus, the internal and the internal and the external globus pallidus. You don't need to know that, but just pointing them out. Now, here's the key thing. Here again, we're going to be like circuit busters, thinking about ways to get over a problem. Spinal cord injuries, what we were trying to get around, and we had, nerves coming to the side. We had chemicals in there to get the nerves across. Here is a diagram of not just the structures, but the circuit of the basal ganglia, but how they're connected up. And this is, can be identified using anatomical tracers, where you can look at where the neurons are going and where neurons, what neurons are projecting to a particular area. So here is the striatum, caudate, and putamen receiving strong input, and this is excitatory input of dopamine, DA, from the substantia nigra. The cortex, including the motor cortex, is also a strong input onto the striatum, an excitatory input. The striatum, the caudate and putamen, projects to the globus pallidus, but this is an inhibitory connection. Remember, we talked about some of these supplementary areas have a lot of inhibition involved. The globus pallidus projects in an inhibitory way to the ventral lateral thalamus that then projects back to the cortex. Here's another key nucleus. Subthalamic nucleus provides glutaminergic, that is excitatory input to both the globus pallidus and the substantia nigra, okay? So just know that I do want you to know this key circuit, what connects to what is excitatory, and what is inhibitory, okay? Know this circuit, but luckily for you, you just get told the circuit, you don't have to figure it out yourself, but what I do want you to think about is what happens when this circuit gets broken. What happens in Parkinson's disease is something very specific. You get death of many of these neurons in the substantia nigra. These substantia nigra cells, there are many, many fewer of them, and there's much less dopamine coming into the striatum. Anybody know what are the symptoms of Parkinson's disease? Shaking? There's a ventral memory loss. We're not going to talk about that part. Anything else but shaking? Any other symptoms that you're familiar with? Yes? Yeah, there are problems with initiation of movement, and there's the palsy that is quite common. There's different kinds, but it's not specifically or strictly associated with the stiffness versus the movement. There are actually inherited and genetic forms of Parkinson's and forms that we don't know about. But that brings up stiffness, lack of facial expression. People can't move their face muscles anymore, and they can't initiate new movements unless they're initiated. So another speaker at our Neuroscience and Society seminar at the Society for Neuroscience meeting was Mark Morris. Anybody know who Mark Morris is? Nobody knows who Mark Morris is? He's a very, very famous choreographer. He's based out of Brooklyn, and his troupe performs all over the world at Lincoln Center. But he came to talk about his program of dance for Parkinson's patients. So it turns out that while many Parkinson's patients have a hard time initiating a movement. They can't start walking, but once kind of somebody gives them a push, they can keep going. Same thing if they start a dance. They could actually do a whole dance and kind of enjoy that movement. And so he has a program at his studio in Brooklyn of dance for Parkinson's patients. So he had some of his dancers come, and there were people with Parkinson's this happened to me in Washington, D.C., and he gave a dance seminar with the Parkinson's patients, and he talked to the rest of the society. So anyway, any other symptoms people are familiar with? Yes?
Difficulty with fine motor movement, like writing or talking.
Absolutely. Fine motor movement is a big, big problem. So remember, I said the striatum is important for direction, amplitude, and initiation of movement. So that means that direction, that you can't like find, the target. A Parkinson's patient will have a really hard time making this movement. They'll kind of get around, or they'll overshoot it because they don't have the right intensity. Or sometimes they can't even do it at all because they can't initiate the movement. Okay, so here's the problem. Damaged cells in the substantia nigra. It happens to be in a brainstem. What would you do? You're a scientist. You are a clinician trying to cure this. What would you do to try and cure this system? You know this person has these motor deficits. Their main problem is damage or many of these cells in the substantia nigra are dying, and there's much less dopamine around. What would you do? What are some solutions that you could come up with? Yes? Inject dopamine. Perfect. In fact, that is one of the solutions. They don't inject dopamine, but they do inject L-dopa, which is the precursor to dopamine. And it turns out if you give the substantia nigra neurons a lot more L-dopa, the ones that are remaining will make more dopamine, and you get a reversal of function. Any other ideas about what you might do? Yes? Inhibiting the reuptake of dopamine. Great. Yes, that is not as common as giving L-dopa, but that's a perfectly excellent suggestion. Any other ideas based on what we've talked about? Yes?
Inject stem cells into the substantia nigra.
Stem cells into the substantia nigra, exactly. And that is one of the techniques that they're doing. It's very controversial, and there are side effects, including seizures that are obviously not good, but some of the patients can improve from that. So that's another excellent idea. So treatments, L-dopa. This is the injecting of the dopamine. Transplantation of fetal stem cells. The other one that we didn't talk about is deep brain stimulation. Anybody heard of deep brain stimulation? Yeah, what do you know about deep brain stimulation?
I don't really remember, I just heard of it.
Okay, just heard of it. Yes?
My grandpa had it.
Your grandpa had it. And was it successful?
Well, it
It helped. Great, great.
a very long procedure.
Uh-huh, yeah.
It helped for a while, yeah.
Great. So this is one of, I think, it's very exciting, very dynamic in the Parkinson's field because they've come up with lots of different things and be able to try different things. Of course, all the transplantation was done in rodents that had, already damage in the substantia nigra, and that was tried. Deep brain stimulation is typically done in the subthalamic nucleus, not always, but you put an electrode put in, and the tip is in the subthalamic nucleus, and you get stimulation of the globus pallidus that gives, again, globus pallidus will then be inhibitory on the circuit and can be very, very helpful. To tell you the truth, people don't know exactly how the deep brain stimulation is working. They've tried different areas to stimulate, and they basically found the one that worked the best. They don't care necessarily about the exact circuit. Neuroscientists care. The clinicians care about getting these people up and walking. And so I just, and it can be actually really, really striking. I think what I'll do is, there are 3 videos that I put up. I want you guys to go home and look at deep brain stimulation 1 and deep brain stimulation 2. They're way too long for us to go today, but it's a great example because you can see how immobile this woman who developed Parkinson's disease in her 50s was, and it takes her through the deep brain stimulation surgery. This is a scary surgery. You don't want brain surgery unless you need it. This is not injection, but a placement of a permanent electrode in her subthalamic nucleus, and with a little radio that will continue to stimulate. And it's actually striking to see how much better she is at her motor movements after the surgery versus before. Not all of them are successful. But what I do want to show you is this video, which just gives you a flavor for the kind of palsy that you see in typical Parkinson's patients. And this is Michael J. Fox, another really, really active spokesperson for Parkinson's research.
Back to the Future was a film that helped define the 1980s and made Michael J. Fox a star. More hit movies followed, as did a string of awards for his TV series like Spin City. Now, though, his future's increasingly defined by Parkinson's disease.
As soon as I wake up, I can't go back to sleep. I mean, it starts going. So I would say that's the toughest part in the morning is to kind of go, well, I'm up whether I like it or not, I'm up now. And the other thing that happens to me in the morning, first thing is I get that dystonia, which is kind of cramping, and it can be very extreme.
In a rare interview on ABC, he spoke about the new challenges from something as simple as using a toothbrush.
Because I have to put the brush down on the countertop, hold it flat, and get the bristles on, because if I hold it, I'll miss it. It's a moving target. But anyway, once I get all loaded up, then I just gotta bring it up to my mouth and then just let it go.
Remarkably, he also revealed that ice skating appears to temporarily block his symptoms,
Kind of
but
like the
only while
dancing.
he keeps moving. There's no cure for Parkinson's, but Michael J. Fox has been fighting to change that. He's gone from actor to activist, campaigning for politicians backing stem cell research, which might offer hope to Parkinson's sufferers. Millions have seen the powerful TV campaign he made a few years ago.
They say all politics is local, but it's not always the case. What you do in Missouri matters to millions of Americans. Americans like me.
Michael J. Fox returns to ABC with...
Okay, so
He
that's
devotes much of...
All right, so just to give you a feeling for what that is like in an everyday setting, we'll continue on Monday with our last lecture before the midterm, Learning Memory 1.